Hello. That's great. Thank you. Sure. It says I'm not allowed to share my screen. Mm-mm. Thank you very much, Swati. I'm having trouble seeing my screen as full screen for some reason. So when I did it yesterday, it worked fine. Okay. All right. Let me try it. Yeah, I'll do that. So are you able to see now the full screen or not? You can. Okay, perfect. Let me just go to the. Okay. Can you see my pointer? Can someone confirm they can see my pointer? Okay, perfect. Okay, so yesterday we were discussing how cancer happens, and there were some very interesting and intelligent questions asked. I would request if you have any such questions for today's sessions, please feel free to type them in, and either I can have a look on them while I'm presenting, or we can have a discussion after 40 odd minutes, 45 minutes to discuss those questions. So please do feel free to ask any questions. No question is silly. Okay, so we were discussing yesterday about this compound called benzopyrene, and we had started going into some details of this compound, which is found in cigarette smoke, among other things. And you can see it is not the only carcinogen, cancer-causing compound found in the cigarette smoke, but there are other carcinogens which are also present. I'm sure many of you are aware of that. Uh, so what happens in cases, I'm sorry, I've not yet introduced you the terms tumor suppressor genes. I'm only calling them good genes. There is a reason for that. And bad genes, which are, I'm sure many of you, especially those who are faculty members, definitely know I'm talking about the oncogenes. So, what happens with the patients who have lung cancer? Their biopsies are taken out, as you know, to define what sort of mutations do they have in the cancer. And it turns out over 50% of the time, the lung cancer in smokers have mutations. What sort of mutations? Mutations that inactivate a good gene. Having a good gene function is always good. It is something that prevents cancer from happening in the first place. If you have inactivating mutations, it simply means the gene is not able to do its function as it was required for it. So this could lead to cancer as well. So when samples are taken out by surgeons in the operation theaters, part of the biopsy goes for immunohistochemistry analysis. Is that what was discussed, Dr. Aziz, in the previous lecture, pathology, just now, Dr. Aziz? Okay, that's fine. Sorry. Is that what was discussed, Swati, in the just lecture, previous lecture? Okay, so the students are aware of that. Okay. So what happens is part of that tissue goes to a pathology lab, and what will happen is, of course, in many countries and as well part of specific projects, the specific genes may be sequenced. And then what was found that in patients of lung cancer, as you can see, this is over 60%, but at least half of them will have mutations in TP53 gene, meaning the TP53 gene is not able to carry out its function properly. And you would wonder what kind of mutations would you find in TP53? And this is not the only good gene. There are plenty of good genes, tumor suppressor genes, which may be relevant to all tumor types, or there may be some which are specific to specific tumors. So what happens, we need to see when benzaparin is incubated with the DNA. Scientists have done these experiments and see what changes occur in the DNA as a result of treatment with this carcinogen, which is found in cigarette smoke. And lo and behold, have a look here. This is benzaparin. In itself, it is not really a big carcinogen, but thanks to our liver, it has become a more carcinogen. And what it does in doing so, it actually incorporates this part onto the DNA, making an adduct. So what you are seeing here is that this benzopyrin is converting, and what you are seeing changes predominantly. This is the addition of hydroxyl group. I'm not sure if there is any pharmacologist on this call. If there is any, would they would understand what is happening here. So the liver, seeing anything coming into the body, recognizing it is not a biomolecule, it's not glucose. Liver knows what to do with glucose. It's not a protein to be broken down into amino acids. This time it sees it as a foreign and tries to make it more water soluble. You can see this molecule benzoparin is not as water soluble as these ones are by incorporation of hydroxyl groups. Why water soluble? Because then liver can get rid of this through kidneys by making them more hydrophilic. They're more water soluble. But in doing so, it creates products which will actually hook up with the DNA, causing these big bulges in the DNA. So this molecule here, which is formed, this is an adduct, and this adduct will cause a big bulge in the DNA, triggering the repair system. Now, as I mentioned on the previous slide, one of the part of the repair system is those good genes, tumor suppressor genes. And you can see the mutations happening in patients are in the very gene that protects us. So what do you think will happen? In this case, and this is not a reaction happening on just one single guanine. There are millions of guanine. Many of them will have this adduct formation due to benzopyrene exposure. And your guess is as good as mine. What is going to happen here is that these adducts will not be removed because the TP53 function is gone in majority of these tumors, right? So what will happen? Something which was not to start with very carcinogenic has been made more carcinogenic as a result of formation of these bulky adducts. And there is a specific repair pathway in a cell to remove these bulky adducts. One of them is a nucleotide excision repair pathway. So do you think making these bulky adducts is good for cells? No, they are not. At least in this context, this is a normal person who sadly happens to smoke. And as a result, what is happening? Changes are occurring in the DNA. But as long as they have their DNA repair system intact, part of which is, of course, TP53 machinery, they will not see this change in the DNA manifesting itself into a cancer. So they still have ongoing changes, but it will not cause cancer to start with. Over time, what you might see that these changes might actually occur in the very gene that protects us against cancer. And if that happens and the cell cannot correct it, this might actually manifest itself into cancer. Does that make sense? Okay, so one of these products shown on your screen is called BPDEG product. This is an adduct. And what has been found is that in DNA, the formation of this adduct can lead to specific mutations. And there are specific hallmarks of these mutations. What are these mutations going to cause? They can change the nature of a base pair from being a GC to an AT, a base pair. We discussed yesterday these mutations which change purine into pyrimidines and vice versa. We call them transversions. But this is not the only thing happening. Uh, what is also happening as a result of exposure to benzopyrene, but also as a result of oxidative stress, we are generating oxidation of some of these nucleotides. So have a look at another. So this is a guanine residue, and you can see benzopyrene product. The product of this reaction, metabolic reaction, has become part of guanine. Where is this guanine? Within the DNA, right? Uh, so what is happening? I know there are some questions. I'll take them right after this call, Hafsa. Um, so what is happening also is have a look here. This is a bulky adduct. This guanine has been hooked up with this product of benzopyrene, but also there are subtle small changes happening within the DNA as a result of oxidative stress. One of these changes is shown on your screen. Again, this is guanine, and what has happened here is that there is an addition of this oxygen molecule. We call it 8-oxo-DG. And this is caused by oxidative stress. And when do you have oxidative stress more? In conditions which make you, for example, benzopyrene exposure through cigarette smoke. I have been unwell with the flu for the past 7 or 8 days. And what do you think is happening in the body? It is accumulating lots of oxidative damage. Do I have to worry about it? Perhaps not, because as long as you have good repair system to take care of defects like this one here, or defects like these ones here, these are subtle chemical changes which can actually alter the structure of DNA. That is what we call mutations. If you don't have them, you don't have to worry about getting cancer. We discussed yesterday, there were 3 changes I was mentioning to you. One of them was oxidative damage to DNA, the other one was purine hydrolysis, and the third one was deamination. We will cover them in more detail. But I did point out that I wonder if we don't have any of these happening in the cell, these changes to DNA, and these are spontaneous changes, by the way. They are happening, many of these changes, like deamination, you would see is happening all the time. These are, of course, caused by carcinogens, but in our body, due to oxidative stress, thanks to our metabolism using oxygen to burn out glucose molecules to produce ATP, this is something we cannot avoid. What we can avoid is this part. We can avoid not being exposed to benzopyrene and a lot many other carcinogens. So I do hope that makes sense. And what is happening here? This is BPDE being generated. This is 8-oxo-DG being generated. And both of these things actually can lead to mutations. What sort of mutations have been found to occur? You can see here, this can change, as I mentioned, a GC base pair into an AT base pair. You would wonder, how is this happening? Why the base pair is changing? And we will discuss this. Have a look at this slide. This is an IHC slide, immunohistochemistry slide. So the previous lecture was based on this. I'll give you, I realize some of you were not there in that lecture, including myself. But what happens here, the tissue is taken out. This is a mouse, by the way, mouse tissue, large intestine. Same thing can happen in human. You can take out the tissue, uh, preserve the tissue in a wax, like paraffin, and then stain it with antibodies. It is one of the most frequently used techniques in biological sciences, especially cancer research. And you can see here, this mouse, A is a control group mouse from a control group, while in B, this mouse was fed something which leads to the cancer being formed in the colon, in the large intestine. If you're interested in this paper, please go and have a look at this PubMed ID. This is the ID of this paper in PubMed. You will find that. So you can see here, this is how normal colonic epithelial expression of 8-oxo-DG looks like. You can see here and there is slight 8-oxo-DG. These are the cells within these crypts of intestine. You don't see substantial amount of 8-oxo-DG in this. But in group B, where the mouse was deliberately treated with a carcinogen in order to make it a model to study cancer, you can see here, this stains very beautifully with 8-oxo-DG, and you can appreciate all these epithelial cells are expressing very high level of 8-oxo-DG as detected by this specific antibody. What does this tell you? That the compound used in this, of course, is causing the DNA to have more changes, which is converting the guanine in the DNA into 8-oxo-DG. And that is setting the stage for cancer. So if you keep exposing these mouse, please have a look at this paper, for a certain number of weeks, eventually many of them will develop cancer. Some of these things we may be having through our diet, unknowingly, we don't know, but many of them we know through our lifestyle, that we are being exposed to agents which can cause this increase in 8-oxo-DG. I'm not saying, like I was alluding before, I'm not saying that if you are not exposed to these agents, you will not have 8-oxo-DG, but that will be very little. And system is pretty capable, the repair system is pretty capable of taking care of this, as long as the repair system is intact. But the moment you overwhelm the system, so now you have a lot of 8-oxo-DG to remove, and consider a situation where these mutations, 8-oxo-DG causing mutations, are happening in good genes, there is nothing to save us from developing diseases like cancer. I hope that makes sense. So I'm going to change, I'll tell you something about these free oxyradicals. So these changes you're seeing here on this slide, benzopyrene, and you can see these adduct formation. This is happening due to cigarette smoke. This can also happen. The 8-oxo-DG can also be found in the DNA as a result of not only cigarette smoke, but also oxidative stress and oxidative damage can lead to this, including benzopyrene and the compound. I'm not for remembering the name, but this compound, which was given to mice, also causes 8-oxo-DG in the DNA. Now, these are the, uh, shall I say, bedrock, these changes in the DNA, these chemical changes, which are making a platform for the cancer to happen. These oxidative stresses are important. But do you know how many diseases, cancer, are treated in clinic? You can give patients the treatment that will cause reactive oxidation species to be formed. What am I talking about? I'm talking about drugs like DNA damaging chemotherapeutic drugs, platinum molecules, platinum based drugs, cisplatin, and so on. What do? They damage the DNA. Do you want the DNA of a cancer cells to be allowed to divide the whole genome? No. So what happens with these drugs? They overwhelm cancer cells by free oxyradicals, which will damage their DNA beyond a point of repair. And these cancer cells will die. You must have heard of the treatments called radiation therapy. And the way radiation therapy kills tumors is also uh, through the generation of free oxyradicals, because that is what it does. High energy causes free oxyradicals to be generated, and that destroys the DNA. So if you see chemotherapy and radiotherapy, in a sense, both therapies are working through a common pathway, and that is through the generation of these reactive oxygen species causing oxidative damage. So if you see here, these mutations, these changes in the DNA, rather, these are happening, they are causing normal cells to go on a cancer pathway to become cancer, right? But you don't want these repair pathways, you don't want these um, oxidative damage, which is happening in the DNA. Uh, you don't want it in order to get cancer, right? But if someone has cancer, you want to overwhelm these cells with this oxidative damage. Why? Because you want to kill those cells. So how do you overwhelm these cells? You do that by enhancing the level of reactive oxygen species in a cell, or ROS. And how would you do that? Either by chemotherapy, most of the time, by radiation. Both of them will lead to oxidative damage, which will degrade proteins, DNA, to a point where the cell will not be able to repair itself. So the same oxidative damage is bad for a normal individual, uh, which, because this has the potential to cause cancer. If someone is being treated with a therapy to kill their cancer, then uh, oxidative damage is what is needed. Giving them drugs like antioxidants might actually be uh, harming those patients. I hope that is clear. So you can see here what is happening. These papers came in the past few years, and you can see here this is overwhelming evidence. One paper, this is, I think, Science Translational Medicine. The reference is here. You can have a look at this paper and have a look. Antioxidants accelerate lung cancer progression in mice. Antioxidants can increase melanoma metastasis in mice. So both of these different papers are telling the same story. If you already have cancer in a mouse model, you of lung cancer or melanomas, you can see the effect of antioxidants. They are not good in this context, because here they are making cancer worse. I think one of those papers, I can't remember on top of my head which one, used vitamin E as an antioxidant. Are we seeing more research coming into this area? Not particularly. Why not? my personal view is uh, that it is what is happening is this is a multi-billion dollar industry buying antioxidants. You come to this country, in fact, in India, you can see uh, anytime you go to a doctor, a drug is given and they give you a stash of some sort of antioxidants, right? Do you need them, antioxidants? And this could make actually case worse for patients who have cancer. So you can see here, this is an alarm for those um, doctors or scientists who think that antioxidants may be good for cancer. I was in a cancer meeting last year across Atlantic, and there was clear evidence presented that from an epidemiological study that patients who were prostate cancer patients who had vitamin E supplement actually have progression of their prostate cancer substantially statistically faster than those who did not have vitamin E supplement. So I think it was vitamin E or D, I can't remember, but both of them actually in many ways may have antioxidant responses. So um, I will stop for 5 minutes to take questions. We have plenty of time. There are some questions, and I think it will make the discussion healthier. Swati, is that okay? Okay, let me just have a look here. Um, so the question I have is Hafsa is asking, Walaikum Salam, Hafsa. With respect to BPDE formation, what molecular factors govern its preferential binding to guanine bases rather than other nucleotides? Hafsa, that is an interesting observation you've made. So this is just one of the examples. There are preferences in chemical systems. I can give you some examples later on, but it seems there is an affinity, higher affinity for guanine to be chemically modified by BPDE, and that could be a preferential effect. The exact molecular factors, I will not be able to pinpoint what they are, but this is perhaps just an example. Others may be modified as well with other chemicals on other specific nucleotides, for example. But it seems the benzopyrene prefers to make adducts through those metabolic changes on guanine residues. Then second question again is in the slide I see higher OXO-DG levels in tumor colonic cells. Does this indicate that the oxidative DNA damage is an early event? Absolutely. So these oxidative DNA damage are indeed earlier events. You can open those mice, sequence the DNA, and may still not find any changes to DNA. So there may not be any underlying mutations at this start of these mice being put on those drugs. But if you leave them for substantially longer time, there may have been few weeks, 16 weeks, I can't remember, then it is possible the cells are dividing. Mammalian cells, most of these epithelial cells will divide once a day, sometimes, usually. And what will happen as they divide, they will accumulate. They will change these damage into a mutation. So DNA damage is a different thing, and mutation is a different thing. But DNA damage is indeed, in this case, needed in order to cause mutation. So 8-oxo-DG is not a mutation; it is a damage. But if it is not repaired, it can lead to a mutation. Sorry. Early event, you're asking, Hafsa, oxidative DNA damage is an early event in tumor development or is a consequence of tumor progression? I would say the first one. Khadija is asking, so antioxidant-rich food should be avoided by cancer patients. Um, I will not say that. And the reason is pretty obvious. Cancer patients are frail. They need good diet. The amount of antioxidants they will get from these will not be very high to have any effect, any negative effects on cancer treatment. A patient being treated, will not have these effects because the sheer amount is very little. Uh, I can tell you there are antioxidants, you will know, found in green tea. EGCG is one of them. Uh, there are capsules being sold. There are capsules of curcumin being sold in places. And what happens? Sorry about that. So what happens is we calculated that in order to make one capsule of EGCG or green tea antioxidant, you will have to, if you don't want to take capsule, you will have to drink 50 liter of green tea at least. That is impossible. But if someone is taking those capsules, might actually be making their disease worse. Uh, I visited some time ago India, and one of my cousins took me to see one of his very good friends. I was obliged. He's very senior to me, so in age, I went, and they were treating his head and neck cancer thanks to good cousin Khanis. So he developed this head and neck cancer, and perhaps the poor thing died by now. And what happened is that they took him to some abracadabra. I'm not sure what it was, herbal clinic, and they gave him a box, and it says curcumin. And I asked him, these must be yellow tablets, and he said, yeah, 5,000 rupees and two of these boxes. Okay, is that what you're having? No, we started treatment and then we stopped because word of mouth, anyone who goes to this person gets treated. Uh, People don't understand cancer therapies don't work like that. And as a result, what happened? You can see this curcumin and other things. They are potential. They have lots of antioxidants, perhaps made the disease worse because that was like big capsules being given to that person. Imagine if this was, you know, in this country, UK or US, people are taking antioxidants left, right, center all the time. Do we need them? What we need is present in our diet. So at least have proper diet to make sure you have everything instead of just relying on enhancing the level of some things which may actually not be good. So as I'm speaking, I may have cells which have mutations, which have changes, but they may not still become cancer because there is a good system to repair those changes. But if I overwhelm my body and have lots of cancer-causing chemicals, what will happen eventually? Cancer. So I hope that answers all the questions. And let's start the session again where we stopped. So you can see from these studies that these papers published in top journals are actually not a good news for people who rely a lot on having antioxidants. I'm talking about general population. They must be taken under. The problem, unfortunately, is, and no offense to doctors on this call, um, we don't know if there are any benefits, but these research suggests there are definitely harm which can be caused by antioxidants if taken artificially, meaning if they are not coming from natural sources like fruits or vegetables, they can actually cause harm. Hmm. So oxidative damage aside, we have this event taking place in the body all the time. We call it purine hydrolysis. And since I started this talk half an hour ago, I guess I probably had billions of these events taking place in trillions of my cells, where the purine is removed from the ribose sugar. It is happening spontaneously in the environment where the DNA is inside the nucleus. There is very little I can do about it. And I don't have to worry about it because hopefully I have a good, and all of us have very good repair system, which will make sure that this DNA, where this is maybe part of a gene or any other area which is gene deficient, this will be seen as a damage and this will be corrected. So just like the adduct formation, BPDE, or 8-oxo-DG formation, this should also trigger DNA damage response, seeing this as being damage. And we call these AP sites. So first thing, oxidative damage happening to the DNA. Um, this is purine hydrolysis. And what you can see in the next slide is deamination. There is very little we can do about it. And what is deamination? You can see there are plenty of cytosines in the DNA, and they have this amino group. If you have a look at the structure of cytosine, you'll find that. So what happens here is this can be deaminated, and as again with purine hydrolysis I showed you on the previous slide, this event is taking place spontaneously, this reaction. So what happens, deamination, as the name points out, there is a removal of this amino group. And you will be surprised, some of you, that this removal of amino group converts cytosine into uracil. Now there's a problem. Uracil can base pair with adenine. That's again not a good news because it will cause mutations. It will change a GC base pair because this is cytosine. If it is part of DNA, on the opposite side of the different strand, there will be a G. So this was GC. But if it is converted into uracil, this can pair up with adenine in subsequent cycles of replication. And that can convert a GC base pair into an AT base pair. That's not a good news. It can cause mutations if it is not corrected. By the way, is there anyone on this call who would tell me what is the target of chemotherapy and radiation therapy? They both generate reactive oxygen species. Maybe you can type your answer if you want to. Um, and we can revisit this in 10 minutes when we conclude this first lecture. For today, what is the target of chemotherapy? You may not find it in the books, but something to think about. Chemotherapy, is there a drug target for chemotherapy and radiotherapy? So, is a cytosine the only one undergoing these deamination events? No, not really. There are others like 5-methyl cytosine, which also undergoes deamination, converting it into a thymine. And again, you can see the cytosine, which 5-methyl cytosine, which was paired with GC as a result of deamination, if not corrected. Uh, so if this is not corrected by the DNA repair machinery, what can happen? This will become thymine, and thymine can base pair with adenine, as you know. So again, in both of these scenarios, these deamination can lead to a GC base pair converting into an AT base pair. And that will be classed as a mutation. So this one and this one, both of them. Now, I would want you must be wondering how these changes are happening. You mentioned, so I mentioned cigarette to smoke, changing the sequence of DNA. How is it happening? I discussed with you these in the previous lecture. Okay. Before I'll take more questions, sorry about that. So have a look at this slide. There are mutations happening as a result of exposure to cigarette smoke, among other things. And can you tell me, I'm seeing some strange changes on my screen. Can you still hear and see the slide, Swati? Okay, thank you. Sorry about that. I think I'm new to Google Meet. I usually use Teams, so sorry about that. Look what happens here. There is a patient who is a smoker, and as a result of this, what you would expect to see in them is the formation of 8-oxo DG. So this highlighted G is now something which has become and had either 8-oxo DG or a BPDE. In both cases, what is this is G which has changed due to oxidative stress. Something similar you would learn can also happen with apurinic sites, meaning where the purine has been taken off the pentose sugar in the DNA. And look what happens here. Uh, DNA replicates, and DNA replication is semi-conservative. So each strand of the DNA, this is textbook biology. Um, you can see here this is strand and this is strand. Each one of them uh, can actually become the template for the synthesis of new daughter cells, right? So those daughter cells which harbor this strand, where this strand, which is not damaged at all, serves as a template. What will happen? You see any mutations? Not at all. So in the new strand, in the daughter cells, there will be no changes. So one of the daughter cells, which is a progeny of this cell with a mutation in one of the strands, sorry, with a damage in one of the strands, this cell will be perfectly fine. There will be no changes in it in terms of the DNA structure, sequence of DNA, sorry. And look what happens here. Uh, if this strand, the upper strand here, DNA, which has the mutation, sorry, I'm saying again the mutation word, which has the oxidation of this guanine, if this strand were to serve as a template for the second order cell, you can imagine what is happening here is that instead of a G, there is a G, there must be a C. Now the system, the repair system will bring an adenine. So the system usually will bring anytime there is a failure and there is the oxidated G, an adenine will be brought. Should there be an adenine here? No. There are pathways which we will discuss in about half an hour, which will see this as a defect and they will correct it, hopefully. So what happens here is this daughter cell is not looking good because it has now accumulated a change in the DNA. It should have been G. There in the new strand shown in red, there should have been a C, and that is not the case. So that cell is not looking good. It's not a healthy cell. And if a repair system is not able to see this as a mismatch, this is a definite mismatch. You can see there should have been a C here. Then this mismatch can lead to first cancer cell being formed. So this is something that can happen as a result of failing to correct the uh, DNA damage repair. Uh, and have a look what is happening here. This is the last slide. And then I'll take more questions and we can have a break of 10 minutes. So what is happening here? 8-oxo-DG, you can see this is formed in a double-stranded DNA. There was a cytosine. And you can see here, when the DNA is replicating, each strand is serving as a template. One strand was fine, as one daughter cell, one strand was okay, and in that cell where this strand served as a template, there are no changes. In the other one, of course, we have now a mismatch which needs to be taken care of. What if this is not taken care of? You can see this adenine comes in here, and if this is allowed to replicate this cell, this monster cell is allowed to replicate, what will happen in the subsequent cycle at this location in the DNA? The base pair, which was originally GC, has now actually become an AT base pair, which is a classical definition of mutation, changes in the sequence of DNA. What about these AP sites? So you have these purines gone. For example, this G is not there anymore. This is happening, as I mentioned, spontaneously. Uh, what is happening here? Again, the AP site, if this strand, the upper strand, were to serve as a template in through DNA replication and the formation of new daughter cells, if this DNA strand were to serve as a template, you can see what will happen here is that there was a G, which is now gone. And what will system bring here is an adenine. And if this DNA with this mismatch is allowed to become repair is allowed to proceed without being repaired, this will change in the next cycle of DNA replication a base pair which was originally GC into becoming into an AT base pair. Again, that's a classical definition of mutation. So with this, I will stop and I'll see what questions are there. And Swati, if you like, you can switch on the speakers as well. So these students and colleagues can ask me questions directly. In the meantime, while you prepare yourself, I will take questions which I see in the chat box. So yeah, I think I've all answered some of these questions already. Uh, Antioxidant-rich food should be avoided by cancer patients? Definitely not. What should be avoided is super high, ultra high doses of antioxidants given as tablets. That is my personal view. Uh, Padmavati, you're absolutely right. Chemotherapy and radiotherapy cause the target is the DNA. So chemotherapy does have a target, which is a DNA replication. Uh, Afi Afzal saying nitrogenous bases of the DNA. Yes, that's correct. Basically, it's a DNA. The target is DNA. But Mawati is saying forms DNA adducts with nucleotides. You're absolutely right, such as cisplatin will make DNA adducts. So those DNA adducts, I just gave you an example, which can make a person sick by developing, leading them to a cancer path. Those DNA adducts are needed in order to kill cancer cells. Fatima Khan is saying chemotherapy interferes. Absolutely right. Uh, Radiation therapy causes direct DNA damage. That's correct. Uh, Direct simply means they will lead to generation of reactive oxygen species, which will cause oxidative stress burden on cancer cells and causing their DNA to uh, get damaged permanently. So Nilofar is asking, why is deamination considered an important source of a spontaneous mutation? Uh, your question has its own answer, Nilofer, because it is spontaneous and it is super frequent. It is happening at enormous levels. Since I started this lecture 45 minutes ago, millions of these changes have happened. There are chemical reactions. You can see within the environment, aqueous environment of the DNA. This is one of the changes happening. And I did not point to you, and perhaps we have time for this, Swati. If I go back to this slide, we all studies right from the high school. Uh, the thymine is present in DNA and uracil is present in RNA. We all know that. Looking at the slide, can you tell me a reason? Um, you can activate your mic if you want, or you can type this question. Those of you who want to take a break, we will meet in 13 minutes again. I'll stay behind before taking a 5-minute break. But one of the answers why you see uracil in RNA and not in DNA, and in DNA we have thymine. You can tell me the answer now, looking at the slide. So Madiha is asking, in the meantime, Padmavati is saying, thank you, no problem, Padmavati. Madiha is asking, sir, so do antioxidant supplement worsen cancer by protecting cancer cells? I will show you some evidence. I've already shown you some evidence from this very strong Science Translational Medicine Journal papers, which suggest actually this is the case. Antioxidants can make the treatment of lung cancer, the progression of lung cancer worse actually, because while you're trying to kill those cells by generating oxidative reactive oxygen species, those antioxidants are going to remove them. You can ask yourself, is it good? Is it bad? And you would see cancer is not something that all of us should worry about. I know the cancer cases are on rise, but in the next lecture, which will start soon, you will learn that actually
Can I answer your
is
question, Doctor? If you,
yes,
if you put a
Dr.
10
Aziz,
pound,
please go on.
if you put a 10-pound reward on them,
On what question? On this?
why DNA has got thymine and RNA has uracil?
this is one of the answers I'm showing them on this screen. So I've given already a clue.
Yes, right. Anybody can answer that question. That's a very simple, fundamental thing, and the answer is right there. I think the post-lunch
But
calorie.
yeah, Dr. Aziz, yeah, I mean, when they're thinking about this, this is quite alarming. I'm sure, Dr. Aziz, you must have seen in US, you go and walk into a pharmacy and all you see is these big aisles of antioxidants where these are not regulated by drug regulators. You can go and buy as many as you want. Do you
Mhm.
really need them? We need to ask some. Uh,
Yeah, but still in US they do test it for the toxicity and other things. So they at least don't have the ingredients, the excipients, which are toxic. So by and large, they remain harmless unless you overdose them.
Okay, so these antioxidants, I was telling these papers which came out in Science Translational Medicine, actually antioxidants are bad
Yeah,
for
I think
cancer.
it depends on the dosage that how much of it are you taking, right?
Yeah, so I gave them an example of EGCG found in green tea. What you
Mhm.
get in the market, pure EGCG, to
Mhm.
have the effect of one capsule, you will have to drink 50 liters of green tea, which nobody does. So you're actually artificially increasing the level of antioxidants in your body. Is it really warranted?
True.
So I think there may not be any questions, Dr. Aziz. Shall we take a 10 minutes break and reconvene?
Yes, absolutely. And we can keep the session live and on. So
Yeah,
please
let's do that.
give the question and you can post
If
it,
you have any questions,
or I can tell you
type in the
microphone's
questions.
on. So if anybody wants to use a microphone instead of typing, that's fine as well.
At least use this as an opportunity to get
Sir,
trained
sir,
to
I'm
ask questions.
sir,
Yeah.
sir,
Yes,
Dr.
Juhi.
Juhi here. Yeah, sir, I learned
Yes, Dr.
about
Juhi. Sorry.
learned about vitamin E, the role of vitamin E, and
Hmm.
the probable role of vitamin E in worsening the lung cancer.
Hmm. That is the evidence from this paper.
Sir, so is there like I have gone through many articles, but they have told that if we are using it for a very high dose, means when the doses increases more than 400 milligram per day, it seems. Is there anything
Okay.
like that? Like if you give it for a long period and also the dose matters.
I think what you're absolutely right. You have a point there. So how much you're giving, what I believe is what is needed is in your diet already.
In
And
that.
it, yeah, I mean, reaching 400 milligram, you said, it would be impossible to, even if you get a truckload of fruits and eat them, you will not be able to, not in case of vitamin E, it may be called liverol or something like that, because it's hydrophobic. But actually, it is quite alarming with the way we have becoming addicted to using these antioxidants as a supplement. That's what I'm saying. May not be a very good idea, actually. And we all know, cancer is the last manifestation of changes, mutations that happen within the DNA, and there is very little you can do many times about this. I mean, the best drug for
No, sir, actually,
prostate
I'm
cancer
doing one.
gives you six
Yes,
months
sir.
to live.
Sir, I'm doing
Yeah,
one clinical,
yeah, Juhi.
one randomized control trial. I'm a
Excellent.
dentist by profession, and I'm assistant
Okay.
professor in Z Dental College, AMU. And we get
Okay.
many cases of pre-malignant lesions, like oral lesions that may
Uh-huh.
convert into malignancy
Yeah.
later on. So
Yeah.
in that, I'm giving vitamin E in a very low dose, like it is 200 milligram,
Okay.
like that, for 2 months, not more
Uh-huh.
than that. Means
Uh-huh.
I ask
Uh-huh.
patient to stick to the regimen for 2 months only. Some people, they when they are not, because it is very cheap also, sir. That
That's right.
capsule, 10 capsules, they it cost not more than 15 rupees. So if they are
Mm-hmm.
not coming for a follow up, they are themselves buying it and having it. So I have told them many
Okay.
times. This is a problem in Indian scenario that I am facing. Even though we are telling them, please, It has to be only for 2 months. Do not go beyond that. So yeah, my sir, I
Yeah.
am totally agree. I usually counsel patient to modify your diet. Get the nutrition from your diet. But then, like, ma'am, it's not possible for us. But what you're talking about, to have like salads, have cucumber, have
Mm-hmm.
tomatoes, like that. So you please
Mm-hmm.
tell me some antioxidant that we can take. It is
Dr.
happening now. How to
Dr.
deal
Gupta,
with such patients?
Dr. Gupta, that is a problem here as well, that people just go and buy, because as I said, this industry is not regulated by drug regulator. So you can go and buy anything you like, but you are
Yeah,
doing
it
an
is
excellent trial.
such a OTC, such a common OTC present. Like you can buy anywhere.
That's right.
So
I think the
yeah,
trial
at
you're
least
doing is excellent, because
huh.
you're seeing these pre-cancer fibrotic lesions probably,
Hmm.
and
Mhm.
giving them before cancer actually happens.
Huh.
So
Yeah,
that
that's
might
why totally.
actually that, that's excellent. That's a very smart idea actually.
Yeah. And I'm doing the biopsy also at the end of the treatments, just to see if
That's great.
any adverse effect has taken place. Till now, no patient has developed any. If the patient has stick to the regimen, I'm targeting only those patients.
No, that's good,
That is.
and especially because it is in the context of pre-cancer, you may be able to extend the window before they develop cancer. And as you rightly said, some of them may not even develop cancer.
Mhm.
Wonderful.
So I'm really sorry for the interruption, but there has been some problem.
Can you not hear me very well?
Yeah, but it's echoing.
I think it is maybe echoing because someone has left their, they're not using microphone, headphones maybe. That's why it is echoing.
Okay, I'll check.
Yeah, can you still hear the echo?
Dr. Juhi, can you
Hello.
turn your mic for a second so that I can hear?
Let's see
Okay.
if it
I'm
is
turning it.
echoing. So testing for the echoing of my voice. I think that's why this was happening. Okay, thank you. Yeah, I would say this is exactly what we need to do. India is such a big country, and even in centers like AMU. Actually, one of my friends graduated from that dental college, the inaugural batch of the dentistry. It's actually a good thing, because with these good cousin Chinese, you see these patients tobacco consumption. The cases are on the rise, and as I mentioned yesterday, this is the last thing one would worry about. There are reports of people not being able to make rational decisions, depression, uh, cardiovascular effects of these consumption of tobacco, good cars, and so on. So this is all congratulations to you and your team, Dr. Gupta, what you're doing through this RCT. Uh, that's great.
Thank you, sir.
Wonderful. Let's touch base with us. You have my email address. Let me know how this trial goes
Sure.
once
Sure.
you
Sure.
have the outcome. Wonderful. Anyone else with any questions, or shall we take 2-3 minutes break if everything is fine with that? Anyone needing a break? Yeah, if you need, we will keep alive. You can ask me questions if you have. So, Dr. Gupta, I met this patient. I don't treat patients, as you know, I'm a scientist. But looking at those reports, they have local invasion, and instead of going and taking the patient to PGI, they preferred giving them tablets of curcumin, hoping that it will treat, because it word of mouth. Many people go to this person, and they were all cured. Cured. I don't know what cure means, but basically, this is quite on the rise in India, which is quite alarming, as I see it.
Yes, sir. That is the problem actually.
And it's very difficult. You can't really, I have relatives, my first cousins, and some families, my uncles and aunties. Some of them developed cancer, died of it, or are in the treatment. It's very hard to make them convince what is the best course of action. They don't realize, they don't appreciate. Anyone tells them, oh, this Baba in Ghaziabad they have a queue of people queuing up. Yes, lots of silly people going and queuing up. They're treating cancer left, right, center, and they go there, pay a hefty amount of money in a hope to get treated, and end up actually getting quite ill and die a very sad death. Uh,
True, sir. There are many cases in which I have advised patient like you should go for biopsy and go for the treatment, but
yeah.
they have not gone for the biopsy. They have gone to some Baba and someone, and then they are returning back with full blown stage 4 cancer, which I have
Goodness.
seen in stage 1 has gone to stage 4.
Okay. Goodness. I hope your trial and those who stick to your adherent people will make a lot of difference. Because you can see these fibrotic lesions, right, before it happens, or?
Yeah. But there will be white patches. There will be
Yeah.
fibrotic lesions will be there. So we are
Okay.
giving treatment and we are doing now. Like whatever the treatment already going on and what I am introducing in that, I'm comparing.
Okay. That's great. I was seeing a report some time ago that they were doing some trial on epidemiological studies of head and neck cancer, actually oral cancer, in people who were going to who drink very hot tea in Iran versus those who never drink any tea. And there was some association I vaguely remember with the hot drinks. So anyway, Dr. Aziz, do we have your permission to crack on if there are no questions? Let's wait for a couple of minutes. Shall we start, Dr. Aziz, Swati? It's being recorded anyway. So yeah, please go on. Okay, that's good. Let's crack on with this then. Okay, so yes, we were talking about these changes, which we have very little or no control over. Why we can have these spontaneous changes in the DNA and how it can lead to permanent accumulation of mutations in the cells. So I think I covered all of this. Okay. And you can see here what is happening here. In both cases, whether this is 8-oxo-DG, which is the oxidative stress causing changes, subtle chemical changes in to the DNA, converting GC base in a double-stranded DNA as a result of mutation, as a result of replication into an AT base pair. And likewise, these apurinic sites, this is one of the most common uh, change in the DNA happening again spontaneously. This can change a GC base pair into an AT base pair. So you can see here, and you can start expecting what will happen to patients who have mutations if you isolate their DNA and sequence it, the DNA base by base, and compare it with the reference DNA sequence from a normal individual. And we won't go into the genomic level, but we will have a look at just one of the genes and have a look what is happening here. This is the data for TP53 mutations in patients who are smokers. Do you see any patterns? Let's start with this one. Have a look what is happening here. You see that there is a higher frequency of Gs converting into Ts. So initially, in normal individuals at specific nucleotide positions within the DNA of this gene, there are Gs. And now in smokers, they are mostly Ts in those locations. Where is it coming from? It tells you exactly that this is caused by the cigarette smoke, which can convert a GC base pair into an AT base pair. So what was initially G, instead of this GC, now there is an AT. And that is why you can actually get a clue of where this mutation is originating from. And this is just one of the carcinogens. There are thousands of different carcinogens, different degrees of the way they mutate the DNA. It is known. The way they can change the structure of DNA may actually influence the diagnosis. If you're seeing these sequences changes in specific nucleotide positions, they may be a result of specific carcinogens, such as this one being more common in smokers versus those who don't smoke. You can see there is a substantial difference between them. Now, yesterday, somebody was asking me, and I am asked this question a lot of time by family and friends back home in India, also in UK. I have some family here too, about people who never smoke still develop cancer, and that gives them a very silly reason to not stop smoking, thinking everyone gets it. Well, there are 25% people who do develop lung cancer, sadly having never smoked, but then we develop many other cancers without any known etiology as well. So have a look at this landmark paper which came earlier this year in none other than published in Nature earlier this year. And this is talking about the mutations which happen in lung cancer in never smokers, short for L-CINS, as you can see. So this paper talks about many different changes that happen in patients who develop lung cancer, but sadly, they develop it despite having never smoked. And what they did, they had a look at the mutational analysis within this paper. Have a look what is happening here. So they collected samples from 871 individuals, since individuals. So they have lung cancers in non-smokers from 28 geographical locations. Some of them are, you can go and have a look at this paper. If you don't get it, please write to me and I can send you a copy of this paper. This should be freely available to everyone anyway. So the most, they've used few things in this paper. One of them is the AQI or PM 2.5, which is one of the measures for air pollution. And they have countries like Azerbaijan in Eastern Europe, Hong Kong in Far East, as examples of very bad AQI. I'm talking about hundreds. And they collected the data, and you can see here that there are increased levels of PM25, which is one of the components, major components, and the DNA damager within the air pollution that can cause an additional DNA damage. So imagine we have all these processes destroying the DNA. They're making the changes, purine hydrolysis, AP site generation, deamination, and someone is a smoker. And on top of this, if you have air pollution, have a look, by the way. This is an excellent paper in this study, and they show what is happening here. You can see these are the increased levels of so what you're seeing here, sorry, I'm trying to coordinate the screens. So you're seeing these increasing levels of this pollutant, and patients experience mutations in DP53. So that's a good gene which has bad mutations, meaning it's not going to function properly. So this is one of the most extensive studies I've seen, which takes into consideration a lot of cases of L-CINS, cancer of lung caused by environmental conditions. It is a very bad news. You can see here these PIK3A mutations, KRAS mutations. So KRAS is one of the drivers. Mutations in KRAS are very common in certain cancers, including lung cancer, and you can see many of them are enriched in a patient. They have other slide in this paper, and they show the mutations which are enriched in Asians patients who are all as these patients, so they're never smokers or excess smokers at least. And you can see here they compared Asian mutations with North American or Europeans, and you can see here in Asians, these again, the mutations which are most common are the mutations in TP53 or this EGFR, which is a growth factor driver, and we will learn today, not today, next lecture, uh, how does EGFR manifest its effect on once it is mutated on the growth of cells, especially with regards to cancer. So very interesting study to look into, and this is quite alarming. They have I was looking at Azerbaijan and Hong Kong. The AQI is really bad in those countries, can lead to actually real time. And this is, of course, association studies, but you can see there are other studies which are referred in this study showing that, in fact, there is a lot of harm which is possible through exposure to environment pollutant to human body, and that can lead to this. This will set a perfect stage for cancer. You can see it this way. So how do you identify what is causing cancer? There are lots of compounds people think might cause cancer. Others, people never doubt this will cause cancer. And there is, on next slide, is very simple test, and this is called Ames test. I will discuss this briefly. Um, uh, Sorry, I'm seeing a question while I'm speaking, and Ilma saying maybe because of AQI 700. Yes, I'm in Delhi NCR. I don't know about Delhi. I used to live in Delhi for some time. Uh, where I'm sitting, the AQI currently is 33. People still complain about London, which is the next city where the AQI is 50. Uh, so coming back to the topic again, have a look at this test. And this is a very simple and very smart test. It can help you identify chemicals which are direct carcinogens versus chemicals which are indirect carcinogens. And what this test involves is using the liver extract. And what you do, you take a suspected mutagen, uh, incubate it in a liver extract, and plate on an agar plate. Now, this is not just any agar plate. This plate does not have histidine in it. Bacteria need histidine to grow. So what will happen if they don't have any histidine on these plates? These bacteria will not grow. Which bacteria are these? These are hist deficient salmonella. They're not going to grow in this extract. So in this test tube, you have three things. One of them is liver extract. The other one is suspected mutagen. And the other thing is the bacteria. In this control tube, you can see there is no mutagen. Nothing grows here, of course. But what you might see that if something has the potential to cause mutations, actually they can remove this defect in the bacteria. There are very subtle defects that make this bacteria not capable of making its own histidine. Just like defects I was showing you, very small defects in previous slides, where a GC, for example, converts into an AT. So you can imagine this as something which has been changed by just one nucleotide. And by doing so, the bacteria, the histidine synthetic synthesis by pathway will be deactivated. By reverting this change, you can activate this pathway. And if that happens, what would you see? You would see those colonies forming on the plate. And that would suggest that this is actually now active. Why liver extract? Because liver extract will allow those chemicals which by themselves are not carcinogens become carcinogens. They can now cause cancer as a result of changes through the metabolic pathways. So if I show you some slides, these are again cartoons. You can see here, this is a background experiment control conditions. You might see some colonies. You always have some contamination. And you can see here establish whether what happens if there was a compound which was a direct mutagen. What will happen is, even without needing the liver extract, you might see a lot of colonies, meaning it has now converted, it has reversed that defect in the histidine biosynthetic pathway, and now a lot of bacteria are actually able to grow and form colonies on that agar plate, which was deficient in histidine. And then there are some examples. Think about these ones. What happens here if this is similar to the background, and you can see this is not a carcinogen. You see more colonies, and compared to the control, of course, this compound is going to be a carcinogen. And if it is going through liver metabolism, what is happening, that this may be the result of this not having the liver extract. It is still a carcinogen, perhaps a weak carcinogen, shall I say, just like benzopyrene. But what happens if you have liver extract as well, then it becomes even a stronger carcinogen. So that test is a reflection. It is directly proportional to the extent, the potential of any chemical to cause mutations. So it does not directly detect carcinogens, but we know cancer is a manifestation of mutations in the DNA. Then many of these chemicals will be potentially carcinogenic. Uh, so that's an excellent test for carcinogens to establish their identity as chemicals with the potential to cause mutations. But how do you test them? The person who tested them first and is credited with developing the first model of cancer is Yamagiwa, a Japanese scientist. And what he did is he took the rabbit and painted it with tar, crude coal tar, the inner ear. And in doing so, they were able to see some changes in the cell not physiology, cell behavior, and identified later on that actually it was making cancer developing uncontrollably, the cells within the inner ear. And this is just a crude coal tar, must have lots of multiple, numerous cancer causing chemicals. So he was the first one to establish this. I was showing you a mouse colon model before, where I showed the accumulation of 8-oxo-DG that was part of other model. You can use those chemicals to induce tumors. Now you will ask me, why do you do such cruel experiments? Especially in UK, it is a big problem with people sometimes coming and vandalizing labs. They don't want any experiments to be done on animals. Little do they realize that the vaccines they all have been vaccinated for, saving them from infectious diseases, actually come from animal experimentation. So there is a big debate. Should we not use animals for experiments? Should we use animal for experiments? There are ways where we can avoid use of animals in experiments. These are harsh, cruel experiments, but in UK, and I'm sure similar things have regulations exist in India, where you can't really give harsh treatment to animals or increase their tumor load beyond certain point. In UK, this is 10% of the body weight of a mouse. So imagine a mouse being a 25-gram, 3 or 4 week old biopsy mouse, 25 grams. You can establish a tumor which should not go beyond 2.5 grams when given through subcut, which is under the skin. So what we have learned are different dots today, that there are chemicals which can cause damage. Do we not get damage in the DNA? We do get damage to the DNA anyway, all the time happening spontaneously. What people are doing by exposing themselves to cigarette smoke, for example, is enhancing those damage, making them perhaps beyond repair. And that will be the first step. A cell will divide, and this cell will divide, and a daughter cell will harbor a mutation. It will not be a mismatch. It will be a proper mutation where at a particular locus within the DNA, the nucleotide has changed. Would that cell become cancer? Definitely, the answer would be, in my view, no. You need those mutations happening to that daughter cell again and again, new mutations, which will make it more fitter, or shall I say, more rebellious, which will not follow the principles of normal physiology and anatomy. And in doing so, the cell will become cancer. So Let's go and talk about this. You can see this slide. I'm not talking about animal migration. Why am I showing this? Have a look. This migration taking place in Europe. These birds go as far as taking a flight from London to New Delhi airport, over 7,000 kilometers, and they carry out this journey. No doubt, you can see some people observing this. Uh, this requires lots of adaptations, lots of endurance exercises to be able to carry out these journeys. And we know these animals are adapted. Not every bird will carry out these migrations over mountains. You can see these birds flying over clouds and mountains. They go as far as Himalayas, or on top of Himalaya, which is I think 8 kilometers or so. They need to be specially equipped. And this is why am I showing this? Because this is something similar happening in cancer cells as well. They need to be extremely fit in order to survive many, many journeys, to allow them to go from one part of the body to other. Have a look at this slide here. These are some of the journeys taken by cancer cells. So what you're seeing here is from a mouse. You can see the liver is completely patched up with melanomas. The cell from the skin's migrating, melanocytes migrating into a lung. You think this might be a simple journey, must have taken uh, a lot of endurance for these cells to have a lot of resilience. Uh, in some ways, I have respect for cancer cells really, with the damage nature gives them.